1. 2. Nitrogen is an essential element in all living organism. It is the constituent of amino acids, proteins, hormones, chlorophylls, and many of the vitamins. Nitrogen is present in the atmosphere abundantly in N2 form, but eukaryotes and many prokaryotes can't uptake nitrogen directly from the atmosphere. The nitrogen enter into the soil from atmosphere by fixation process. Then from the soil plants absorb it, and from plants it moves to animals and fulfill the requirement of all living beings. 3. Plants competes with microbes for the limited nitrogen that is available in the soil, so nitrogen is a limiting nutrient for both natural and agricultural ecosystem. The nitrogen cycle showing relationship between the three main nitrogen pool: atmosphere, soil and biomass. Steps of N2 cycle. 1. N2 fixation. 2, 3, 4, 5. Uptake and assimilation of nitrogen by plants. Aminification. Nitrification. Denitrification. Nitrogen fixation. Process of conversion of atmospheric N2 into nitrogenous compounds NH3, NO3, is called nitrogen fixation. Nitrogen fixation is of two types: 1. Abiological, physiochemical. 2. Biological. Abiological nitrogen fixation. Abiological nitrogen fixation. Physiochemical N2 fixation is further divided into two types: A. Electrical. B. Industrial. A. Atmospheric or electrical. In nature, lightning and ultraviolet radiation provide enough energy to convert nitrogen to nitrogen oxides, number 2, N2O. B. Industrial N2 fixation, in the presence of high pressure, temperature and catalysts. Nitrogen and hydrogen combines to form ammonia, NH3. This ammonia is used in the formation of chemical fertilizers. Biological nitrogen fixation. Diazotrophy. Conversion of atmospheric nitrogen N2 into inorganic nitrogenous compounds like NH3 by living organisms is called biological nitrogen fixation or diazotrophy. L. Nitrogen-fixing organisms. Diazotrophs. A. Asterisk. Free-living or symbiotic. Eubacteria. Azotobacter, Bajornichia, both aerobic, and Clostridium, Rhodosperlum, both anaerobic. Cyanobacteria, blue-green algae. Nostoc. Anabina, Tolopothrix, Orlocera, etc. Symbiotic diazotrophs or N2 fixers. Paspalum notatum gra have loose symbiosis, associative symbiosis with Azotobacter paspali in roots and Azospirillum with wheat, malls, sorghum, etc. Usually Azospirillum fix the atmospheric nitrogen in free-living condition. Both Rhizobium and Frankia live freely in the soil but fix nitrogen only when in symbiotic association with host plant. W enzyme nitrogenase catalyzes the conversion of atmospheric N2 to NH3. It possesses two units. Unit first is MOF protein and unit second is Fe-S protein. Nitrogenase is extremely sensitive to oxygen. So to protect it from oxygen, leguminous nodules contains an O2 scavenger called leghemoglobin (LHB), which combines with O2 to form oxylehemoglobin (LHBO2). Legumeglobin is pink or red in color, globin part synthesized by plant and heme part synthesized by bacteria. Asterisk. 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 Many cyanobacteria capable of fixing nitrogen are filamentous and contain thick-walled cells called heterocyst. These are the sites of nitrogen fixation. Heterocyst lacks oxygen-evolving photosystem II, thus do not evolve O2 and protect nitrogenase. Biological N2 fixation involves contribution of following genes: I. Nod gene of host plant. E. Nod. Fix and NIF nitrogenase including factor genes of nitrogen-fixing microbe. Asterisk. Asterisk. The atmospheric N2 is reduced by the addition of hydrogen atoms. The 3 bonds to nitrogen atom N equals N or dinitrogen are broken step by step, and ammonia NH3 is formed by reduction of N and then ionization of ammonia NH3 to form ammonium ions NH4-1. Ferredoxin helps in electron transfer. N2 fixation requires 3 components: I. A strong reducing agent NADPH2 FADH2 NADH2 from photosynthesis and respiration. E. ATP from respiration. In symbiotic fixation from the respiration of host cells. Nitrogenase enzyme. Rhizobium legume symbiosis. Rhizobium is a free-living, gram-negative non-sporulating, aerobic and motile rod-shaped bacterium. Rhizobia are more prominent in the rhizosphere of leguminous plants. Asterisk. Nodule formation. Nodule formation involves a sequence of multiple interaction between rhizobium and roots of the host plant. Principal stages in the nodule formation are summarized as follows. I. E. Rhizobia multiply and colonize the surroundings of roots and get attached to epidermal and root hair cells. The root hairs curl and the bacteria invade the root hair. An infection thread is produced carrying the bacteria into the cortex of the root, where they initiate the nodule formation in the cortex and pericycle of the root. IV. Then the bacteria are released from the thread into the cells, which leads to the differentiation of specialized nitrogen-fixing cells. The nodule thus formed establishes a direct vascular connection with the host for exchange of nutrients, T. Uptake and assimilation of nitrogen by plants, FATEOFAMMONIA. Reductive amination in organic NH3 absorbed soil as NH4+ reacts with a ketoglutaric acid to form an amino acid glutamic acid. This process known as reductive amination or amino acid biosynthesis. A ketoglutaric acid plus NH4 plus NADPH plus H plus. Glutamic acid plus H2O plus NADP. Transmination transfer of amino group from glutamic acid to other keto acid is known as transmination. This is process of formation of other amino acids from glutamic acid in plants, catalyzed by transaminase enzyme. Glutamic acid plus OAA aspartic acid plus acetoglutaric acid. Glutamic acid plus pyruvic acid alanine plus acetoglutaric acid. Transportation of assimilated N2. In plants, transportation of assimilated N2 through xylem occurs mainly in form of amides, glutamine and asparagine, especially in leguminous plants. Amides are more stable than amino acids and possess high nitrogen to carbon ratio 2n-5c in glutamine, while glutamic acid possesses 1n-5c.